everyone. In this video, we will study about the concept of the heterosis. You all are amazed to know that, or maybe you all are curious to know about the hybrid concept. We all generally say that hybrid have the superior performance over the parents. Now, why this takes place? Now, why this process is there? The superiority of F1 over and above to their parents. It is majorly because of the heterosis. So, in this video, we will understand the concept, how the hybrids have the higher performance. Just take the example, if we have the two broad or the distinct pure lines, the F1 which we will get will be of this genotypic constitution, having the 50% genotype from one parent, 50% from the another parent. So both these are the homozygous line and this is the heterozygous. And the performance of this heterozygous is more than this homozygous or more than this homozygous parent. So this is known as the superiority. Okay, so first we will understand the concept of the heterosis. So heterosis term was given by the Schull in the year 1914. It may be defined as the superiority of an F1 hybrid over both of its parent in terms of yield or any other character which we are considering as an input. Just take an example, if a plant is having the height of 20 centimeter and this is the condition of the genotype, and we crossed it with the dwarf genotype having the height of 5 centimeter, and we got the F1 with this heterozygote, and if it is having the height of 30 centimeter, so you can say that it is performing over and above the one of the parents. So it is because of the heterosis, or this process is because of the heterosis. Okay, it may be also the condition that instead of 30 centimeter, the height of the F1 is 2 centimeter. Okay, so again it is superior than the dwarf parent, but it is in the negative side, but it will also consider as a effect of heterosis. Okay, so generally heterosis is manifested as the increase in vigor. We are just saying that it is 20 centimeter and this height is 30 centimeter. So basically the increase in the vigor of that particular character, or maybe the size, growth rate, or you have other characters. Okay, some cases hybrid may have the inferior to the weaker parent, which I just told you. This is also regarded as heterosis. Okay, sometimes heterosis is estimated over the superior parent. Like if we will take the character of height, so this is the superior parent as compared to this parent, so it is the superior parent, and F1 is performing over and up to the superior parent. Then it will be referred as heterobeltiosis. Okay. Then is the hybrid vigor, the concept of hybrid vigor or the definition of hybrid vigor. We generally use the term hybrid vigor in the definition of heterosis. More or less, these two words are synonymous. It is just like the word we use the heredity and then we use the word inheritance. Both are used for the transmission of character from one generation to the another, but heterosis is generally used as a process, hybrid vigor, when we are defining a particular character. Okay, so again hybrid vigor will also describe the superiority of the hybrid or the F1 over the parent, while the heterosis describes the situation overall. So basically heterosis word is used for the situation overall. Now the features of heterosis. Ultimately, because we are making the process because of our own interest. What is our interest? We want the economic output, that is majorly the yield. So increase in the yield, or we can say the increased yield. This is the first and the foremost feature of the heterosis, and generally we use the hybrid, and we used to say that hybrids have the higher performance than the other varieties. So this is due to the feature of, or we can say this is because of heterosis. Then increase in the reproductive ability. Hybrid generally exhibit the increase in fertility as well as the reproductive ability, which is ultimately required for the propagation. Then increase in the size and the vigor. Definitely, if the economic size of a crop is increasing, definitely we will have the benefit. Greater resistance to the disease and insect and pest, because we are making the crosses between two diverse parent. Two diverse parent means they have the broad genetic base, and we are crossing these two broad genetic base into a one hybrid, or we can say one line. So definitely it will have the effect of both the lines. So ultimately it will have the greater resistance to the diseases and pests. Now there are some factors which ultimately affect the heterosis. One of the foremost factor is the mode of pollination. Heterosis is higher in the cross-pollinated species because there are chances of having more and more combination, because any of the pollen can fertilize any of the ovule, while in the selfing same pollen is fertilizing same ovule, so there will be no genetic variability. Okay, so the chances are higher in cross-pollination. Genetic diversity of the parent, it also depends. If the parents are not diverse, then there will be no heterosis. Similarly, genetic base and then the adaptability. So ultimately, the thing which I described you earlier, so this is the condition. Okay, so next is the exploitation of heterosis. How we will use the heterosis? So first and foremost thing is making the hybrid varieties. How we will make the hybrid varieties? When the F1 generation from a cross between two or more pure lines. Pure line word is generally used in the self-pollinated crops. Inbreds, inbred word is used in the cross-pollinated crops. Then the clone is used in the asexually propagated. Okay, and other genetically dissimilar population lines is used for the commercial cultivation. It is known as the hybrid varieties. Okay, it means generally, as we have discussed, we are making the cross between two parents or two homozygous lines. Ultimately, we are getting a heterozygous, or we are calling it as a F1. So it is a hybrid. So the commercially cultivated hybrids are generally of two types. One is the intraspecific, another is the interspecific. Intraspecific means if we are making the crosses between the lines of a particular species. Just for example, Triticum aestivum. Okay, if we are making the crosses among the lines of Triticum aestivum, then it will be called as intraspecific. But if we are making crosses between Triticum aestivum and Triticum durum, so this will be a interspecific because the species are different. Okay, and if we are making the crosses between the lines of Triticum aestivum, then it will be called as a transspecific. Okay, like interspecific is genetically different genotype of same species. These genotypes are different, maybe differing for a particular character, but they are from the same species. So there are three type of methods from which we can make the crosses. One is the single cross hybrid, that is we are crossing lines or two genotypes and getting that. So this is the single cross hybrid. Then is the three-way hybrid, three-way cross in which we are crossing two lines and then we are making F1. Then after making it inbred line, we are crossing it the C, another line. Then the F1 which we are getting, it will be a product of three-way hybrid cross. Okay, then is the double cross hybrid. Double crosses, we are making cross between four different lines. F1, from this we will also get the F1, then we will make them homozygous, so that again we can make a cross between these two homozygous lines, and ultimately the F1 which we will get, it will be known as the double cross. So it is a cross of two single cross. Again we are making a cross between product of two single cross. This will be known as double cross. Okay. Then is the interspecific cross hybrid. Interspecific means when we are making the crosses between two different species, we are calling it as an interspecific. Just for example, Gossypium hirsutum, Gossypium barbidens. You can see the Gossypium genus is same, and hirsutum and the barbidens species are different. So this is the interspecific hybrid. Then the manifestation of the process can be synthetic variety as well as composite variety. Synthetic variety is generally when we produced a variety by crossing some of the inbred lines in all possible combination. Okay, so this is the synthetic variety, but we have some limited inbred lines. Okay, this is the main feature of synthetic variety. While in the composite variety, we are generally mixing the seed of several phenotypically outstanding lines and encouraging the open pollination. But what we have done in the synthetic variety, we have just taken some of the superior inbreds and then we are making the crosses in all possible combination, and according to their combining ability, then we are maintaining them by open pollination. Here we are not making the crosses, we are directly growing and encouraging for the open. This is basically the difference. Here we can understand more. Number of inbred lines are generally 6 to 8, but here we have selected some of the superior seeds, so it may be up to 20. GCA, general combining ability of the parental line, is tested because we have made the crosses in all possible combinations. Here we have not made any combination. We just give the opportunity for the open pollination. Synthetic varieties performance can be predicted, but the composite variety performance cannot be predicted. Neither they are reconstituted. Synthetics can be reconstituted, and the seed replacement can be done by 5 years. Here we have to replace the seed earlier by the year 3 to 4. Then the the next concept is of the inbreeding depression. So this concept is just opposite to the heterosis. In the heterosis, we were discussing, or we were talking about the superiority of F1 over the parent. Here we are saying the F1 is producing, or the progeny is producing poorer than the parents. That's why we call it as a inbreeding or the consanguineous mating. It is a mating between the individuals that are related by the descent or the ancestry. Descent or ancestry means the pedigree is same. When the individuals are closely related, like brother sister mating, that is also known as a sib mating, or we can say the half sib mating. This is the degree of inbreeding is high. Now, what do we mean by the brother sister mating, sib mating, or half sib mating? Means if the parents are same, okay, if we are making the cross between two same lines, so the individual will have the same kind of variability, or the variability will, after a long period of time, it will they will become the homozygous. So there will be no variability. And half sib means when the one parent is common. Okay, so it will be known as half sib. So the degree of inbreeding is high. Highest degree of inbreeding is achieved by selfing, because we are making a cross between a same genotype. So definitely there will be no other variability. Inbreeding depression may be defined as the reduction or loss in vigor. In the heterosis, we were discussing about the hybrid vigor. Here we are talking about the loss. And the fertility as a result of inbreeding, it can be calculated as performance of F1 minus performance of F2 divided by the performance of F1 into 100. By this way, we can calculate the inbreeding depression. Now, the heterosis breeding. So, like we have seen, for the commercial exploitation, we are making the hybrids, we are making the synthetic, we are making the composite. So, this is the commercial exploitation, or we can say the result of heterosis breeding, where we are basically exploiting the process of heterosis or the characteristics of the heterosis. Highest percentage of outcrossing possibility is there. Rural biology should be as such, so that large scale production of hybrid seed with the less expenditure is possible. So ability of male style and the self-compatibility system make this process easier. Okay, so this is about the heterosis breeding. So this is all about the heterosis concept. Hope you will understand the complete compatibility. System make this process easier. Okay, so this is about the heterosis breeding. So this is all about the heterosis concept. Hope you will understand the complete concept after taking the test also, as well as after studying the notes also. For any doubt and query, you can ask in the live class. Thank you everyone for watching this video. Hello everyone. In this video of plant breeding, we will discuss about the important, or we can say the practical topic, that is the crop improvement. Although the previous topic which we have studied are also practical, but like the concept of heterosis is basically the theoretic, or we can say something else. But the crop improvement is the thing which we generally practice in the field. Like you can see the picture, these are the breeders. They are basically assessing the crop, or they might be making the crosses. So in this video, we will discuss about the crop improvement, specifically in the self-pollinated crop. I hope you all have seen the videos in which I cleared that this concept of what are the self-pollinated crops and what are the cross-pollinated crops. Now you might be thinking, what does this word mean? Crop improvement means what kind of improvement? Okay, so crop improvement is nothing. It is a term used, or we can say this term we generally use when we are upgrading a variety. Okay, or you can say when we are enriching the genetics. Genetics means we are incorporating the genes through the continuous breeding, that is also known as the conventional. This is not by the genetic engineering, it is by the conventional breeding. We are making the process again and again, and we are incorporating some useful characters into the variety which are not present earlier. Okay, so we are like just have an example. Just take an example. There is existing variety which is not yielding more. Just take the example, like there is a variety X producing 40 quintal per hectare. It might be the variety of paddy or rice, or the variety of paddy. It is producing the 40 quintal per hectare. Now, due to the use of the variation that is present in the population, we crossed with some other varieties, and now the new variety Y, which is newly created, and it is producing the 50 quintal per hectare. So what we have done, we have basically improved the performance in the terms of yield. Okay, and this is not only in case of the yield. There may be other character like the disease resistance, like some crops having the incidence of some specific diseases you may call as the yellow rust or black rust. So we incorporated the rust resistant gene in the variety, and now the variety become the resistant. So this is again a type of crop improvement. Means new variety is not at all susceptible for the diseases. Okay, so this is the crop improvement. Similarly, other traits are also there. There may be some herbicide resistance, or there may be some other economic factors. Even we can enrich them for the nutrients. Like you have heard the term biofortification. We are enriching the wheat for the iron and the zinc. Okay, so this is also comes under the crop improvement. But here the important thing is we are improving our crop with the help of conventional breeding. Okay, now what is conventional breeding? We will study in this video. I hope you have got a basic idea about the crop improvement. And this we will discuss only in the cell-pollinated crop. The very basic and the first thing is the mass selection. This is the basic and the common method we generally used to do in our daily routine also. So mass selection is the oldest method of crop improvement. In this, what we generally do is individual plants are selected on the basis of the phenotype. Means, like if we want some plants, or we can say if we want a variety having the this particular height. Okay, and but in a population, in a field, we have some plants like of this height, this some of this height, some of this height, some of this height. So we will purposefully select the specific plants having the same type of phenotypic characters. Phenotypic means the physical appearance, because we can't check the genome of each and every plant. So individual plants are selected on the basis of phenotype from a mixed population. Mixed population means every type of individual, or we can say the segregating population also, every type of individual is present. These seeds are bulked and used to grow in the next generation. Seeds are bulked means we have selected some of the plants. Now the seeds are harvested and they are mixed, and they are used to grow in the next generation because they have the same phenotypic appearance. So they may produce same type of phenotype in next generation also. But before that, we have the two requirement of selection. There should be variation in the population. Means if you want to select this kind of height, so it should be present in the population. Okay, so it should not like there like the population having this much of height, and you are looking for the this much height of plants. Okay, because this type of trait is not present in the population. So the variation must be present in the population. Variation means, like simple, the English term variation means every individual have something different. Okay, so that differences should be in the population. Then variation must be heritable. Okay, it should not be due to some agronomic, or we can see the some of the specific condition. Like just take an example, if you will not provide the sufficient urea or the nutrients or the irrigation, plant growth will be stunted. Okay, plant growth will be stunted, plant will be height will be very limited. And if you will select, and basically why this height is very small, because you didn't provide the urea, nutrient or the irrigation. Otherwise it could have grown about this much of height. So if you will harvest the seed and grow for the next generation, definitely if that plant will get the urea, nutrients and the irrigation, it will attain this much of height. So this is not the heritable trait. It is due to the what we can say the environmental conditions. It is not because of the genetic trait. So that variation must be heritable. These two are the important requirement of the selection. Okay, so it has the two major application. This you can understand why we generally do this thing. One is the improvement of desi or the local varieties. You all know that the desi or local varieties means already existing varieties are there, but due to the mixing, or we can say the different type of plants, the variety is not producing much. So what we can do is we can select some of the plants which are yielding better than the other plants, and those seeds can be mixed, and then that can be grown in the next generation. Because this is a self-pollinated crop, we can do this. Then the purification of existing pure lines. Pure lines means having the same genetic, we can say that same genotype in all the plants. So they should perform equally. But if there is some mixture, so through the selection, we can easily select those plants which are similar in the population, or we can say we can rogue out. Rogue out means removal of the plants which are off types. Okay, like this is the population of a common heighted plant, but like if some plant is performing like this, so we can rogue out. Rogue out means we can you know uh, just
Uproot.
uproot this plant from the population. This is the basic thing. Okay, so these are the two basic applications of mass selection. Now we'll understand what is mass selection. So mass selection is, as I told you, this is the population. These points are presenting the plants, individual plants. So this is the population. So from a variable population, variable means there should be, variable means the plant should have the different type of phenotypic appearance. So what we will do, 200 to 2000 plants with similar but desirable traits are selected. They should be similar, but they should have the desirable trait for which breeder is looking, or for which we are looking. The seed from the selected plant are composited. Composite means this can be mixed in a container. Then the composite seed is planted in a preliminary yield trials, means as the breeder has selected some of the desirable plants, and these desirable plants are now performing well, or we can say from the existing population, so they can be planted for the preliminary yield trial along with the standard check. Standard check means the existing high yielder variety, or we can say performing better for that particular character which is in the question. Okay, then the phenotype of the selected plant is clearly evaluated in the preliminary yield trial. Then the promising selections are evaluated in the coordinated yield trials. Now what is the coordinated? Means we are growing that particular crop in different locations, at several locations. So this is the coordinated trials. You might have heard about this term, ACREP, All India Coordinated Research Project. So what they do, they simply all the agriculture universities, the seed which they are producing, they used to grow, they grow that seed in different locations, so that to understand the performance of that particular variety which is not variety, that is basically line after release, that will be known as a variety. So it outstanding raised as a new variety. Seed multiplication is for the distribution. So this is the process of mass selection. Nothing has to be very difficult. You have to just select the plants, and then those seeds is composited and then grown. Now the merits and the demerits, that is important. And one more thing is the genetic composition. Many times it is asked that mass selection population has which type of genetics, like it is homogeneous. Homogeneous means every plant will be of same type. Okay, and homozygous.
Zygous.
Homozygous means this is the homozygous condition. Just take an example for a character, capital A, this is the homozygous condition, capital A, small a. If the alleles are different, then this is a heterozygote. Okay, this I have already discussed in the basic genetics, so you can understand this concept from there. So this is homozygote, and this is heterozygote. Okay, zygote means zygotic condition. Then homogeneous means all the plants having the same type of zygotic condition. Then the merit is the good method for the improvement, as we discussed, most stable in performance because all the plant having the same genotypic and the phenotypic appearances. It provides the good protection against the diseases because we have already selected those which are promising. Then simple and quick method of crop improvement. You can see it is not the labor intensive. You have just select the plants and we have to grow them again. Then what are the demerits? Still there are demerits. Selection is based on the phenotypic. We don't know about the genetics, and as I told you, it may be environmental also. It may be some plants getting higher nutrients, higher irrigation, so they are performing well. It may be due to any reason. Okay, and specifically in the cross pollinated, although we have studied it for the self pollinated, there is no control on pollination. So there will be the repetitoriation of the variety because but the condition is constant. There will be no segregating population. Okay, but the cross pollination will lead to the segregation in the coming generation. In cross-pollinated crops, large number of plants have to selected for bulking because small sample will lead to the inbreeding depression. This we will discuss in the crop improvement for the cross-pollination methods. Okay, the produce of variety developed by mass selection is less uniform than pure line. Okay, then pure lines. These are less uniform than pure lines because these are not pure lines. These are performed identical. Okay, the method is applicable for both self and cross. Now, the second method is pure line. In the mass selection, we were not discussing about the zygotic condition, but here we are discussing about this condition, that is the pure line. Pure line means these are the superior phenotypes selected based on the phenotype only, but having the pure line. These are the pure lines, or we can say the condition of homozygote. So 200 to 3000 plants are selected for the superior phenotype. Then individual plants progeny grow. So if the condition is with this, so there will be no segregation. Any type of combination. If it is making, if we are making cross with this plant with this plant because this is a self-pollinated, so the progeny will be like this. But if this is the condition of one parent and because we are making the self pollination, so then we will get this type of genotype in this type 25%, 50%, and then 25%. So this will be the segregation. So this we will understand by the individual plant progeny. So after the selection of individual plants, what we will do? We will do the individual row of all the harvest from the individual plant. So if the progeny of the plant of the particular plant are performing similar, then that will be counted as a pure line. Otherwise, progeny will be rejected. Okay. Then we'll take it for the preliminary yield trials, then multiplication yield trials, then coordinated research trials, and then finally the superior progeny. As compared to the check, if it is doing well, then it is released as a variety, and then seed multiplication for the distribution. Now again, merit and demerit. So merit is maximum possible improvement over the original variety because it is a pure line. These are extremely uniform because genetic content is same. Definitely, ultimately, phenotype will be also same. But due to the extreme uniformity, the variety is easily identified in the seed certification program. So this is again a merit. But now the demerit is do not have the wide adaptation and stability because all the plants are similar. If this will be the condition, there are two different kind of alleles, but here are the two, both are the similar. So they have the less adaptation and the less stability in the population. One potential pest can eradicate complete population, and it requires more time, space, and expensive yield trial. You can see, we have to perform this individual plant progeny test also. Then improvement is dependent on genetic variation present in the original population. It also matters. Breeder has to devote more time in the pure line selection than to the mass selection. Now this is the pedigree selection. Now this is different. Here we are doing, just different from the mass selection and the pure line selection. What we are doing here is, we are making the process. Now what do you mean by the making process? Here we have two parents. We have identified two parents which are having the desirable traits. We are making cross so to get progenies having the traits of both the parents, and the two are desirable. So what we will do in the first generation, we will make the process. Then those seeds, or we can say the F1, first filial generation, will be space planted and harvested in bulk. Then again, then we'll not make the cross, but what we'll do, we will harvest the crop, and then we'll space plant it, and so we are selected. So we are also making the selection, and selection from the third year. This you have to understand. So in the pedigree selection, what we are doing, we are making the crosses in the first year. Then in the second year, we are just growing them, and then from the third year, we are making the selection. Okay. Then individual plant progenies are space planted, and then superior plants are selected, and we are also looking, because these are self-pollinated crops, we have to understand whether they are homozygote or not. Then similarly, after these individual plant progenies, then we'll grow again the individual plant progenies, then it will go for the pre-varietal trials or the yield trials, then for the coordinated yield trial, then will be released as a variety. So it takes more time than the mass selection and the pure line, but this is better than the pure line and mass selection, because in earlier two methods we are using the variation that is present in the population, but here we are creating the variation. We are creating the variation. The variation is not available in the genetic pool. We are making the crosses and we are making the variation. Now the merits and demerits is it is provide the maximum opportunity for the breeder to use his skill for the selection of desirable variety from the segregating generation, because it is a good population, number of plants are more. Transgressive segregants means the outstanding progenies or yield and other quantity may be recovered in addition to the improvement in specific trait. Okay, so along with the specific trait, we can also get some other transgressive segregants. The breeder may often not able to obtain the information about the inheritance qualitative from the pedigree. Plant progenies with visible defects and weaknesses are eliminated at earlier stage because we are doing so much of selection for the superiority. Okay, so then the demerits are maintenance of the pedigree record, because what is the pedigree means we are making all the records of parents, and this is a tedious and time consuming and labor intensive job. Okay, selection among the within a large is laborious and limited number of process for the breeder. Success of this method depends on the skill of breeder, means the selection of parents, then after making the cross, the selection of progenies. This will lead to the ultimate success of the breeder. No opportunity for the natural selection to influence the population because we are doing everything. Now the bulk method. Bulk method and pedigree method is similar, but here the difference is similar to the pedigree. We are making the crosses between the desirable parent, then the F1 we are getting and harvested and planted in a bulk. Then from F2 to F6 means that from the third year to the sixth year, what we are doing, plants are planted and commercial seeded and harvested in bulk, means no selection. We are doing no selection. Okay, but in the pedigree, what we were doing, we are doing the selection. Here we are not doing any kind of selection. Okay, now from the F7, plants are space planted and superior planted, harvested separately. Superior plants means we started the selection. Okay, so this is the difference. In the pedigree, we were doing the selection from the F3 generation, or we can say the third generation. Sorry, from the F2 generation or the third year. Here we are doing from the 8th year and the F7. Then similar to the previous method, just things are doing. Here the merit is natural selection increases the frequency of superior genotypes. By providing this condition, commercial seeded, we are checking them for the natural selection. Large population is grown, so the greater chance of isolation of transgressive segregant than in the pedigree method. So this is the merit. Okay, again, these are the different merits and particularly suited for the small grains. In case you can read, this is not very much different. Then the demerit is natural selection become more important only after F10 and bulking may have to turn up to 20 means 21 years longer than the time taken in the pedigree. This simply means that double time. Short-term bulks are useful for the isolation of the homozygous line, but natural selection has little effect on the bulk population. Okay, opportunity for breeder to exercise this skill, but the important thing is this is very time-taken process. Now the back cross method. It is little bit different. Here we are making the crosses, but here one is the recipient parent, one is the donor parent. Donor parent means donor parent is a parent from which we need only one or few genes for a recipient parent. Means this variety is performing well, but this in the due course of time or maybe due to any reason it may become prone for a particular disease. So what we will do, from this variety having the resistant gene for that particular disease, we will make the cross. Although this variety is not performing well as compared to this variety, so we need only one gene. Only one gene needs to be transferred to this parent. So this is the recipient parent, and this is the donor parent. So what we will do, we'll make the process. We'll get these things, smaller and capital R. Then what we will do, again we'll back cross with this recipient parent. Again and again we will cross it with the recipient parent, so that the rest of the thing will be of recipient parent only for that particular character, for which we are looking, and that too is dominant in the donor parent. Only that will remain in the population. Rest things are similar to the pedigree population. The merits are genotype of new variety is nearly identical with that of recipient parent, except for the gene transport from the this do that, like this way. That is the demerits. New variety cannot be superior to the recurrent parent except for the character transfer. So this is the condition. Only the particular trait which we have transferred, that will only that will be only visible. Otherwise, the yield and other things will be similar. So this is all about the methods for the conceptual query. Hello everyone. In this video, we will discuss about the crop improvement in cross-pollinated crops. So in the earlier video, we discussed about the crop improvement in self-pollinated crop, and in that video, I discussed about meaning of crop improvement. So if you haven't watched that video, so you can watch that video prior to this video. Okay. So in this video, we will discuss about the cross-pollinated crops. So as you all know, cross-pollinated means it is a population, or we can say the field of plants. You can take the example of maize field. It is a cross-pollinated crop, so there is equal chances of getting crossed with another pollen. Cross improvement in the cross-pollinated crop improvement in cross-pollinated crops. So there are two main thing. One is the population improvement, and another thing is hybrid and synthetic varieties. These two things are nothing. These two things are the ways to make a new variety, or we can say new population, which is performing better than the existing. Okay, so this is the crop improvement. Now it is very clear because we have already discussed in the previous video. Improvement means making a new variety which is performing better than the existing or the previous variety. Okay, so in the self-pollinated crops, we discussed about the selection. Then we discussed about the crossing. In the crossing, we discussed about the pedigree, then bulk, then backcross. Okay, so here in the cross-pollinated crop, what we have to do? We have to go for the population improvement. There we didn't use the word population. Why we are using the word population in the cross-pollinated crop? Because you have to understand that this is the basic, or we can say the natural way of pollination. So we cannot check. So what we can do is we can improve the population as a whole. Okay, now what do you mean by the population improvement? Means a variety in an open field. Okay, I will explain it later. So just in case of the population improvement, mass selection or its modification are used to increase the frequency of desirable alleles, thereby improving the characteristics of the population. So what we have to do is, here the important thing is, in the population improvement, we have a population means we have a field of maize. Okay, we are doing a mass selection. Now what do you mean by mass selection? As we studied in the self-pollinated crop, we have to select some of the superior phenotype of our interest. Of our interest means of the particular character for which we are looking, and its modification. So what we will making, we are increasing the frequency of desirable type. How the frequency will increase? Because if you will select the suitable type of plants, then there will be chances of crossing between only those plants. So the frequency of desirable allele will be more. So this is the basic thing, is the pollinative population improvement. Then another thing is hybrid and synthetic varieties. So in hybrid or synthetic, what we are harvesting, we are using the F1 generation, or in the synthetic we are using generally more than 2 or 3 generation. In case of hybrid and synthetic, a variable number of strains selected on the basis of their combining ability are crossed to produce a hybrid population. Okay, now here the word is combining ability. You all know that hybrid is basically the summation of character from each of the parent. But how we will understand that if a parent is doing better for a particular trait, another parent is not doing better, then how those combination of those you will express in the F1. So that is the combining ability. Both of those characters are exhibiting, or we can say how they are evolving and how they are expressing themselves in their progeny, or we can say in the F1 generation. So basically that is the hybrid and synthetic varieties. Now the population improvement, as I discussed, it is already grouped into general classes. But in the self-pollinated crops, what we were doing, we are selecting some plants and we are making the process. Okay, here we need not to make the small process, because plants are already cross-pollinated, so there are chances of n number of processes. So what we can do is selection without progeny testing, and another one is the selection with progeny testing. Okay, Now, what is this progeny testing and why we are having this particular approach? What we are doing? Population improvement. Population improvement means for a particular cross-pollinated variety. Selection with progeny testing means we are selecting some of the superior phenotype from the population. Okay, and as you know that the population is cross-pollinated. Okay, so we have selected. Now what we will do, just take an example. You have selected a particular plant from the maize field. Okay, there are so many plants. You selected one of the maize plants. What will you do? You will harvest, and from the cob you will get the seeds, and all those seeds will be planted in a next year in a row. So this is the selection with progeny testing. Okay, what we are doing, we have selected the parent, and then we have grown the progeny, and now we will see the performance of this parent. Okay, because we have selected, we are assuming that it will perform better. So now we will check on the basis of the performance of progeny that whether this type of plant is suitable or not. If it is performing, all the progenies are performing according to the requirement or according to the selection, then it is okay. Otherwise, we will discard this. So this is the selection with progeny testing. We are testing the progeny. We are estimating or we are calculating the worth of parent based on the progeny. Similarly, selection without progeny testing means we have selected some of the superior plants from the existing population, but we are not going for the progeny testing. Okay, it means on the basis of the phenotype we have selected, now we will go for the population improvement or for the IUI initial varietal trial, and then for the multi-location yield trials, and finally for the variety. Okay, so then selection with the progeny testing, this is the main thing, and this is very important because we can see, because these populations are already segregating. There are combinations are there in the every generation. So progeny testing is one of the best methods. Then under this selection with progeny testing, we have the following methods: progeny selection on year to row. This is basically this method which I just told you, that from a particular plant we will raise a complete. Similarly, if you have selected this particular plant, you will all the progeny in another, then you can easily compare, and then you will get the idea how these are performing in the field and how their progenies are performing. Then is the line breeding and recurrent selection. These three things are there. So progeny selection we have already discussed. Now we'll discuss the line breeding and the recurrent selection. Okay, so the progeny selection, this method developed by the Hopkins, and this is the basically gist of progeny selection. Now steps are: number of plants 50 to 100s are selected on the basis of phenotype superiority. They are allowed to open pollinate because this is a cross-pollinated variety. Progeny rows are grown from the selected plants. So from the each selected plant, what we will do, progeny rows will be grown. They are evaluated for the desirable character, or we can say the character that is in the question for which the selection is done. From the superior progeny, several superior plants are selected based on the phenotypic character. Just take an example. If you have selected some plants you have selected, and then you make the row of each of the plant. Okay, then based if these two are performing good, this is not performing, so we will discard this, and from these two better performing progeny, we will again select some of the plants, then we will allow to open pollinate, and plants are harvested separately. Then small progeny rows are grown, and the process of selection raising progeny rows is repeated till the superior population is obtained. Because what we are doing, we are doing selection, or we are doing everything in a cross-pollinated crops. Then we have to have the superior population, not a superior plant. Okay, so maybe 2 or 3 selection cycle. At the end of superior plant, the superior families will be selected and composited to produce a new variety. This is the progeny selection. Okay, then is the line breeding. Progeny selection, you can easily understand, based on the performance of progeny, we are evaluating the parent. Okay, so this is the basic concept. Then is the line breeding. Line breeding is again, it is a kind of, synonymous to multi-lines also. So it is a variation of progeny selection in which one or several cycles of selection is carried out based on the progeny test. Now what is this saying? It is a variation of progeny selection. Variation in the process of progeny selection. Now, what is there at the end of selection process? The last selection cycle, the superior lines of progenies are composited to produce a new variety. what we are doing, we are selecting some of the superior lines based on the phenotype and we are compositing and releasing as a new variety. But you all know this is a cross-pollinated crop and they have the different genotype as well as genetic constitution, because these are the segregating lines. Just take an example, if this is a population, so in the next year definitely we will get these type of three genotypes. These both will perform similarly. Okay, so we have the chances of maximum these type or maximum these type. It may be depend upon the condition. Okay, so if this is the condition, so this we generally studied as a line breeding, and it is known as line breeding in contrast to the progeny selection, where the superior plant from superior progenies are composited at the end. The merita and demerits are more or less same. Now the third method is the simple recurrent selection. Recurrent means recurring, okay, again and again. It is just similar to the progeny selection but having the some different aspects. Again, there also we were doing the selection, then we testing the progeny, then we are doing the selection, then progeny testing. So that was the cycle in the progeny testing. Here in the simple recurrent selection, simple original population is there, the plants are grown. What the farmer will do, he will select the phenotypically superior plants. Then these plants will be self-pollinated. Okay, seeds harvested separately, seeds evaluated, superior seeds retained. This is the original selection cycle. This is the basic selection. Now what we will do, individual plant progeny is planted, and these plant progenies are of the self-pollinated selected plants. Okay, now what we will do, we will make the crosses but not allowed to open pollinate. Okay, we will all possible intercrosses made based on the studies, or we can say the combination. An equal amount of the seed from all intercrosses will be composited. Okay, then composited intercross seeds planted. So these seeds will be again planted. Then the first selection, recurrent selection cycle. Okay, again this will be repeated as many times as time required for the population improvement. Then there are three different methods under this, three different types under the recurrent selection. One is the recurrent selection for GCA, another is the recurrent selection for specific combining ability, and then is the reciprocal recurrent selection. Now what you have to understand is, the important thing is, in the recurrent selection for the general combining ability means we want to check the two cross-pollinated varieties of the same crop, how they are performing when they are crossed with each other. Okay, in general we are checking, we are not looking for a specific character, or you can say we are checking it for the yield character or normal how it is performing in the field. So this is the general combining ability. But if you want to talk about a particular trait, like for a particular disease, just take an example in the rust resistance, or we can say the stripe rust, or any diseases of any of the cross-pollinated crop, you have a certain or the particular trait. So then we will call it as a recurrent selection for the specific combining ability. We will check in lines for this particular character. So now what is the difference? Now you got the meaning, GEC and the specific combining ability. So difference is, in this method the tester is from which we are testing with the broad genetic base, that is the open pollinated variety, synthetic or the segregating generation used for the evaluating the lines of GC. Lines is which we have selected. These are the lines which we have selected. Now we will test with, or we can say we can check with the open pollinated variety or synthetic or segregated. Then is the recurrent selection for the specific combining ability. Here the tester is, procedure for the recurrent selection for the SC is identical to the recurrent selection for the GC. Everything is same, but the tester difference is on the tester. Tester here is inbred, or we can say the narrow genetic base, because we are looking for a particular trait. So what we will do, we will have a particular, or we can say the pure line, or we can say inbred. Generally pure line we use the word in the self-pollinated crops. We use the word inbred in case of cross-pollinated crop. So we will cross with this. Okay, so this will become the specific combining ability. Then there is a reciprocal recurrent selection. Okay, the objective is to improve the two different populations simultaneously. So we have a two population. Earlier we have only one population. These are the plants grown. Okay, this is a one type of variety. This is one type of variety. Both varieties have some of the beneficial character. What we will do, we will pollinate. We'll make the crossbreeding. So this will act as a tester for this. This will act as a tester for this. Okay, this is the simple reciprocal recurrent selection. So this is all about the crop improvement in cross-pollinated crops. So generally in the examination, there is not much questions from the plant breeding section, but if they may ask from this particular topic only. Okay, so this is all about today. So